Hello students, this is the 6th PPT for chapter Biotechnology. In continuation with the topic, the process of recombinant DNA technology, I'll start with the 5th step, ligation of the DNA fragments into a vector. So, now what we have to do, ligation means joining of the DNA fragment into a vector. So, before that the vector DNA and the source DNA from which we will get our gene of interest GOI are cut with the same endonuclease so that we get the sticky ends which can join each other. These are then ligated or joined by mixing them and adding enzyme DNA ligase. So, DNA ligase is the enzyme which helps in joining the sticky ends. The 6th step is insertion of recombinant DNA into the host. So, as I have explained you earlier, before insertion the recipient cells are made competent. We treat the recipient cell with some chemicals to make it competent to receive the foreign DNA. Now, if the recombinant DNA carrying antibiotic resistant gene, example ampicillin. So, for example, if the recombinant DNA carries one antibiotic resistance, for example ampicillin is present, is transferred into the host cell also becomes ampicillin resistant. So, when we will grow or transform these cells on agar plates containing ampicillin, only transformants will grow and others will die because our host cells were ampicillin resistant. For this, we also call this gene as selectable marker. The next step, 7th step, is culturing the host cells on a suitable medium at a large scale. Now, once our cell is part of the host cell, now we want to culture it on a large scale. The transformed host cells are grown in appropriate nutrient medium and the DNA gets multiplied and expresses itself to form the desired products, means the protein what we want. The 8th step is extraction of the desired gene product. The cells having gene of interest, gene of GOI, can be grown on a small scale or on a large scale. On small scale, the cells are grown in cultures in laboratories, and then we manually with the help of the instruments we can isolate the product on small scale. On large scale, the cells are grown in continuous culture system. For this purpose, we use bioreactors. In bioreactors, we can culture 100 or 1000 liters of biomass at a time. And there is one system in the bioreactors by which from one side we can insert the sample and from other side we can take out the product. So, it is a continuous culture. Bioreactor. A bioreactor is like a large vessel where the different cells such as human, plant or animal cell are used to culture new biological products. In bioreactor, we provide optimum conditions like temperature, pH, substrate oxygen level, etc. All is in optimum conditions. The bioreactors are of two types: simple stirred tank bioreactors and sparse stirred tank bioreactor. This diagram is from your NCERT book. The first diagram is of simple stirred tank bioreactor. You see, it is like a big vessel and it is having one propeller in the center which keeps on rotating inside, and acid base pH controller, steam for sterilization, foam breaker. So, this is how a simple stirred tank bioreactor look, and we need to insert the culture and then we can grow it on in large amount. The second one, B, is sparge stirred tank bioreactor. The difference is that in sparge stirred tank bioreactor, we sparge the oxygen bubbles from outside. So this helps the level of oxygen inside, and this results in good products. So these two diagrams you should draw, and you should learn how to draw these diagrams properly. And then the last step is downstream processing of the products as finished products ready for marketing. So downstream processing is a sequential step in which the isolation, purification and preservation of final products are done before it is marketed. We can make capsules or syrups or whatever is the final product we require, that all steps are part of downstream processing. The final product is formulated with additives like preservative, colors, followed by clinical trials. So this completes the chapter Biotechnology Principles and Processes. So dear students, if you find difficulty in any of the topic, you can WhatsApp me personally and I will help you in this regard. Thank you so much.